Okay, to digest protein, what do we need? We need pepsin. Remember, in the stomach, we have an enzyme called pepsin that can help us to digest protein. So, first of all, you need to know which cells secrete pepsin, which cells secrete hydrochloric acid. So, in here, chief cell secrete pepsin, and then parietal cells secrete hydrochloric acid and intrinsic factor. We'll talk about what the function of intrinsic factor later. But just keep in mind that parietal cells secrete HCl, hydrochloric acid, and intrinsic factor. Chief cells secrete pepsinogen. So what happened is that for the question that I raised in one of the earliest in one of the previous videos is that how can pepsin be secreted by the stomach cell without digesting the stomach itself? Because pepsin is an enzyme that will digest protein, and the stomach wall is basically a muscular sac. So it turns out that the cell, they store pepsinogen instead of pepsin. So pepsinogen is an zymogen or precursor protein or inactive protein. So the chief cell going through transcription translation, and then it makes the pepsinogen in the vesicle, in the secretory vesicle, so that it will go through exocytosis, and then the secretory vesicle will merge with the cell membrane, so that the pepsinogen can be released to this we call it crypts or gastric pits, and then pepsinogen will go into the gastric lumen through this crypts or gastric pits. And then what activates pepsinogen? It is the HCl, hydrochloric acid, the strong acidic environment will activate pepsinogen into pepsin. If you remember the basic biology, protein, they are susceptible to pH and temperature. Under extreme pH or high temperature, protein will undergo something called denaturation. pH or acid or strong acid can denature protein, meaning that it can change the conformation or the structure of the protein. When the structure of the protein or the conformation of the protein is changed, then most of the time the protein will lose the function. If the protein is enzyme, then the enzyme can no longer speed up the reaction of a specific substrate, because the structure is changed, the substrate can no longer fit into the active site in the enzyme. Similarly, in this case, the HCl is not actually activating, it is actually denaturing the pepsinogen. But in this case, when the structure of the pepsinogen is changed, it actually becomes activating pepsinogen into pepsin. So most of the time when protein going through denaturation, it will deactivate that protein. But this is one of the few cases that denaturation of pepsinogen actually activate the protein into pepsin. So HCl, hydrochloric acid, will activate pepsinogen into pepsin, and after we have pepsin, it will go through the positive feedback. So pepsin will also activate pepsinogen to become pepsin. So remember, there are two things that can activate pepsinogen. One is HCl, another one is the active form pepsin. Pepsin can go through the positive feedback loop to activate pepsinogen to make more pepsin. And then pepsin is the active protein that can break down protein into peptide. And I also mentioned that there's a thin layer of mucus coated on the lining of the stomach. This mucus is to protect the corrosion of the stomach lining from hydrochloric acid. One of the reasons is that in the mucus we have bicarbonate ion dissolved in the mucus so that it can neutralize the hydrochloric acid. So remember, chief cells secrete pepsinogen, parietal cells secrete hydrochloric acid and intrinsic factor. So how does a parietal cell secrete hydrochloric acid? Let's take a look at it in the next video.